Here we are at, let's move that out of the way, Part 3, Tissues or Histology, page 123. And in medical school, one of the things that we did in the first year was look at slides in the library. We would get a little room, we'd get the slide projector, we'd get slides, and we'd toss all these slides up and take a look at them. We used to call them throw-ups because there was so many of these things that we thought that we might throw up. But at any rate, the intensity with which we were doing that was based on pathology. So we were studying pathology, not just anatomy, but we were studying anatomical pathology. And we came across cancer, lung cancer, and it often was, the lung cancer was squamous cell carcinoma, a fairly common form of cancer, common to, say, Humphrey Bogart, typically at the age of 48, presenting with basically a huge, how shall I say, this maybe down here at the base of the trachea, where the trachea divides ultimately to primary bronchi. Right around there would be something the size of a baseball or a softball that was causing a rusty sputum. You'd go down, you'd get a little sample of the tissue, you'd pull it out, and it didn't look like this, right? Instead, it looked a lot like this. And why is that? Is because there's two stages to cancer. The first stage is referred to as transformation, and the second stage is malignancy. And so what you would end up with is you would go to where you would normally be finding pseudostratified columnar epithelium, and you would be going down here, you'd be taking a little sample. You'd also be noticing that your patient is one who is without cilia, and they're having to have smoker's cough all the time. That is to say, they don't have the tissue anymore that actually expels particulate material. Okay? And then you have to wonder, whoa, why is that? And it's because what happened is that there was irritation to the system. And so what did the trachea do? The trachea looked at its nearest neighbor, the esophagus, and basically redifferentiated itself as stratified squamous. And you can find that in people who have been exposed to years of cigarette smoke. And students will ask me, oh, can you recover from that? I says, yeah, quit smoking. And actually the programming is very strong, and you do tend to return to the original programming. But you can disrupt the programming and end up with this type of tissue in the wrong place, in the trachea or the bronchi, and end up with squamous cell carcinoma. So that was our question to our professor, was, hey, how come we have squamous cell carcinoma when we were expecting to see carcinoma of this tissue, right? And instead of seeing carcinoma of this tissue, we saw carcinoma that was based on this tissue. And the professor took the time to let us know that if we looked carefully at the transformed tissue, that the nuclei were actually clear. That is to say, a transformed tissue is a tissue that's so disrupted and so confused that it has opened up its entire genome. All 46 chromosomes have come undone and naked, and the cell is feverishly looking for another place to be, a different way to differentiate. And so in the case of the trachea and smoke, cigarette smoke, or you can consider coal mining, the same kind of deal happens with coal miners, with the type of cough and black lung that they develop, is that the cellular structure here has become so irritated that it chooses a tissue that's good at abrasion. So this is a good tissue for abrasion. You can swallow pills. You can not chew your food well and still swallow it, and it goes down, and you can abrade the surface here and remove a little bit of the surface cells. Take one of those big calcium tablets or one of those big multivitamin tablets, and it just sort of scratches the whole way down. At any rate, the idea here is that this is a tissue that's good for abrasion. Okay, whereas this is a tissue that's good for removing small, tiny little particulates of the air that can be found mixed in with the air. Okay, so you can see why somebody who smokes has a trachea with this type of tissue, also somebody who's in a coal mine. Okay, so I just wanted to have that little detail transformation. So the second step, of course, after you've transformed and you've opened up the genome, is that the promoter portions of the oncogenes are available. Oncogenes are actually not intended for cancer, even though they produce cancer. Oncogenes are actually for the fetus and for the embryo to grow quickly, right? So we have these special growth proteins and growth genes called oncogenes. They're supposed to be turned off by birth. But if the tissue is transformed and the DNA becomes naked due to transformation of the tissue, then we can get a malignancy. So it's a two-step program, right? Step number 1, transformation. Step number 2 is the turning on of an oncogene, and that's malignancy. Step 1, step 2, transformation, malignancy. Okay, so now you know all about cancer. It took you a few minutes. Okay, so cancer is really complicated, so there's more to the story. But at any rate, now we're on to this next situation here. We're actually looking at the urinary bladder, which, as you know, if you drink water in proper amounts every day, is that it's filling up and emptying. That is to say, its interior volume is changing all the time. And so as a result of that, we have a special tissue here lining the inside, and it is a stratified tissue. In this case, instead of being flat at the surface, it's sort of puffy at the surface, and we call these the dome cells, or at least that's how my professors referred to this. And so the dome cells here are usually big puffy cells that overlie more than one underlying cell. So the daughter cells go through their, how shall I say, mitotic divisions, and they percolate up here in the stratified realms. And then when we get to the surface, we actually get some big dome cell that forms. Okay, and that these can be stretched out. So obviously, when they're on the slide here, there's no urine in the urinary bladder, so there's no stress on the cells, so they're not flat. But they could be. And so if this were a tissue that was actually in a living urinary bladder and there was a lot of urine in the bladder and these were being stretched out, you can imagine that they would be able to sort of have sort of an elastic effect, as it were. Okay. Then that sort of covers our epithelial tissues, just the way you like it. We do have glands, both endocrine and exocrine. They make a brief little notation here that endocrine glands are ductless glands. That is to say, endocrine glands produce an effective molecule. It may be one such as estrogen, or it may be one such as growth hormone. That is to say, it has an effect upon the human body. Okay, and usually at some distant location. And so that means that they're going to go into the blood. So we say that there are ductless glands, that is to say, they go directly from the cell population that produces the molecule directly into blood. And that's how they get around. Okay, and when they find their receptor, they bind to the receptor of that particular cell in that particular tissue. And there's a result. Okay, so it's possible for breasts to grow. It's possible for shoulders to become larger and wider in the male. It's possible for many things to happen. It's possible to have increased metabolic rate if you have thyroid hormone available. Okay, so that's the deal with endocrine glands. Then we have something called exocrine glands, which are the ones that have ducts, and they deliver something to a specific spot. Okay, so we're familiar with, for instance, sweat glands or even sebaceous glands in our hair cells. We might be familiar with salivary glands that produce saliva, right? So there's many glands throughout the body, and we're going to be producing various substances that are useful in certain locations. The stomach, of course, has its gastric glands, and the small intestines utilize the products of the pancreatic glands for enzymes, right? So we have a lot of glandular activity, and they rely on ducts to get their export to certain locations.